Since we are now at the arranged time, so I'll start today's webinar. So, hello everyone. I'm Bingzhou Han. I'm currently a research scientist in GemPharmatech. Today, our topic will be the genetically modified mice models and their applications. So I believe everyone of our audience admit that lab animal studies have really driven our biomedical research improvement, especially in the last century. So from genetics to cellular biology to developmental biology to biomedical research in every aspect. So among all the lab animals we use, I think mice is one of the most important one, since it has some really good advantages that cannot be easily substituted, like mice has a really close genetic relationship with human, and it is the currently most developed and applied mammal model. And there also have been a very good and advanced developed application of genetic tools and other advantages are listed here. So as Professor Shirley Tillman already tell us that the mouse sequence will be the Rosetta Stone that will help us interpret the human genome. So during the development of the last century, we have multiple ways to obtain mutant models in mice. Traditionally in genetics, we have two directions or two main approaches to get the mutants. The first is from the forward genetics ways, like we obtain the spontaneous mutations, the phenotype causing spontaneous mutations when we maintain the animals, or we just add some random induced mutation by using physical or chemical inductions to disrupt the genome, like radiation, chemicals like ENO, or using transposons to do gene trap strategies, and then get the individuals or lines with certain phenotype. After mapping and mechanism study, we could complete the forward genetic study approach. And then as the development of the genome sequencing and the genome targeting modification strategies, people have now really come to the reverse genetic strategies to do targeted mutation of the gene or DNA sequence of interest and generate novel models by an embryonic stem cell based targeted mutagenesis strategy or in the nucleus mediated strategies, like using the Zinc-finger nuclease, TALEN, or Crispr Case based strategies. So here's a brief timeline of the development of the genome editing technology. Since Watson, Crick, and their colleagues discovered the DNA double-strand helix structure in the middle of the last century, then 30 years later, after we obtained some of the gene sequence and gene cloning informations, the first generation of the targeted modification strategy based on the embryonic stem cell by homologous recombination was developed, and then comes the development of the engineered endonucleases like Zinc-finger, TALEN, and the currently most popular CRISPR-Cas strategies, which also obtained the Nobel Prize of Chemistry in 2020. So I believe most of our audience is familiar with the CRISPR Cas9, so I'll briefly go through of the principle of how we apply it. So the CRISPR Cas9 system is mainly composed of two components. One is the protein nucleus, is Cas9, and the other one is the RNA sequence, which is responsible for the target specific, the sequence specific targeting of the nucleus to guide it to the special sequence by using the base comparison approach through the RNA and the DNA. And every DNA endonuclease, including Cas9, will result in the generation of a DNA double-strand break in our genome. And then our endogenous cells repair this double-strand break mainly through two styles of methods. One is end joining, including a conventional or a macrohomology mediated methods. And without donor, the end joining will result in uncontrolled indels, which will generally cause frame shift mutations and will disrupt the endogenous gene function. And also we could give donor to achieve a orientation non-specific insertion. And in the other manner, which is homology-directed repair, which is generally occurred in dividing cells, we could simultaneously introduce donor sequence, like either in a single-strand manner or in double-strand DNA donor manner, containing flanking by the sequence homology to the sites flanking the double-strand break, which is called the homology arm, to generate DNA replacement or targeted integration in a precise manner. That means mutation-free manner. So compared to the most traditional embryonic stem cell-based methods, CRISPR-Cas9 have certain advantages, as we can show here. For a general ES cell targeting approach, we start from the construction of the vector and the culture of the embryonic stem cell, and we use antibiotics like neomycin or puro to select of the cells that contains the insertion, and then we inject the selected embryonic stem cell into the blastocyte, get the chimera founder, and then obtain the heterozygous mice by outcross of the chimera to the background mice. Generally, this strategy takes like 14 to 16 weeks through this culture process, which is really time consuming. However, when compared, let's see in case of we use CRISPR-Cas9, we only need to prepare the Cas9 and guide RNA and some vectors if we need to do knock-in. So this process could be as short as 2 to 6 weeks, and then we inject these components into one cell stage embryos, and then we can get the founder chimeric mice, which is not cellular chimeric but genetically chimeric, and then we also cross this founder with the background mice, and we can get the heterozygous. And this time process seems equal in these two strategies, we can actually save a lot of time in this pre-injection process. Now, you know that time is money. Time is very vital for our both scientific and industrial study. So this is the reason that why now in the nucleus like Cas9-based methods are more appreciated in these days. So this is the end of the background. In the first of this webinar, I want to briefly introduce the genome modification strategy of mice in GemPharmatech. So briefly, the consequence of gene modification could be divided into these parts. The first is the knockout, including our conventional knockout and the conditional knockout, which means that we could disrupt the endogenous gene or its function in a conventional or conditional like time or tissue-specific manner. And then we could do knock-ins to put a exogenous coding sequence or regulatory sequence or a point mutation or reporter gene or epitope tags into the genome in the site we want. And also we could combine different or multiple knock-ins and knockout together to do complex modifications, like we can simultaneously do knock-in in multiple targets, and we could humanize an endogenous mice gene and to substitute it with a human homologous gene. And last, we can do transgene to achieve overexpression of gene fragments, either in a controlled manner or in a random manner. So in our facility, the commonly engineered mouse genetic backgrounds are the common strains we use, like B6, like BioBC, like NOD, and others. And also we could do this modification in the SD rat strain. So first, let's look at the conventional knockout strategy by Cas9. We actually, in these days, we have multiple ways like vital essential exon deletion or generation of a frame shift knockout in the upstream of the gene or in a essential exon, or design two Cas9 target sites upstream of the 5' untranslated sequence and downstream of the 3' untranslated sequence to delete the whole coding gene. So these three strategies can all generate a functional null allele. However, the application of these strategies is dependent on the gene structure and the purpose we want. We need to choose different strategies in different occasions. Conventional knockout by Cas9 is relatively easy these days, and easy to achieve and easy to obtain. However, conventional knockout cannot treat certain difficult questions, like when the whole body knockout leads to an embryonic, prenatal, or neonatal death of the animal. Or in other cases, when the mechanism study, when the phenotype is very complicated, like when there's abnormality in multiple tissues or organs, or if there's any possible secondary effect caused by crosstalk between different cells or tissues, or if there's any genetic composition issue, or if the gene function will be different or will not be identical during different life process, like when it is different during embryonic development and then the maintain of the adult life. When this case comes, conventional knockout cannot help us to solve these problems. So that's why we need to do conditional knockouts. So basically, the first step of generating conditional knockout by Cas9 is to introduce this called flux or frogs strain by introducing two LOXP or ROX sites flanking one or multiple essential exons of the target of the gene of interest using CRISPR Cas9 based knocking strategy to generate this flux of frogs allele. And then in the next stage, when we cross this mice, which is homozygous of the flux of frogs allele with a Cre or Dre transgenic mice, and then the two sites will recombinate and delete the intron, delete the coding sequence, like the essential exons between them, and then generating a functional null allele. So here's a brief figure that interpreting the principle of conditional knockout by a tissue-specific Cre driver crossing with a homozygous mice containing the flux allele of this gene. And then after the crossing of these mice together in an individual, and in the cells which the Cre is expressed in this tissue cell-specific manner, Cre will mediate the recombination of these LOXP sites and generating a homozygous inactivated Y gene in this tissue or certain cells to generate a tissue-specific knockout. And also in that case, scientists have developed this time-specific or inducible Cre line by fusing the estrogen receptor domain to the C terminus of the Cre recombinase. And this is a mutated form called ERT2. And without the ligand of this ERT2, it will maintain the Cre protein in the cytoplasm by interaction of this ERT2 to heat shock protein 19. And then, however, if we introduce the strong agonist or ligand of this ERT2, which is tamoxifen or 4-hydroxytamoxifen, into the individual, the coactivation of this agonist to ERT2 will release this protein from the cytoplasm, and this protein will be then introduced into the nucleus of the cell, causing the genome, the LOXP sites on the genome to be recombinant and delete and excise the essential exons to generate a knockout, to result in knockout. Thus, we could achieve the inducible time-specific knockout by using this Cre-ER. So to make this more vivid and easier to comprehensive, I'll give a brief case study using this PTK1 gene as an example. So PTK1 is well known to involve in development of all kinds of muscle, including the skeletal muscle, the smooth cell muscle, or our cardiomyocytes. So if we want to study the function in tendon development using a mice model here, we will briefly think to generate a conventional knockout of this gene and study its function by observing the phenotype and study uncommon the underlying molecular mechanism. However, if we do this, we will obtain no homozygous knockout of this gene, since PTK1 is essential in all muscle development, including the cardiomyocytes, and thus its loss will cause embryonic lethal, since the heart will be not functional in the embryos. So if I really want to study the tendon development, the function of PTK1 in tendon development, I'll use this SCX promoter-derived Cre and combined with the homozygous PTK1 flux allele to generate a conditional knockout in the SCX positive cells, which is the tendon cells. And in this case, the embryo will develop normally and be born normally. And then we will see and examine, we have the result of the tendon-specific PTK1 KO knockout, and then we could observe the phenotype of these mice. And also, we already know that PTK1 is essential in cardiac development, but what is its function in adult heart? Like if we do a microinfarction surgery to the mass, if it is, and we want to see if it is functional in the heart injury response, or it may contribute to the regeneration capacity of the heart. So in this case, we can use this myosin heavy chain 6 promoter drive CRE-ER to cross with the PTK1 hormone to combine with the homozygous flux mice of PTK1. And then because we don't give any induction in the early development, the PTK1 function will be retained in the embryo heart, and it could be developed and be born normally. It could develop and be born normally. And then in adulthood, we could inject tamoxifen in the mice and lead to the cardiomyocyte specific or MYO6 cell positive cell deletion of the PTK1 and the loss of function. And then we could do the coming experiments to see its function in adult heart maintain or injury response. Other than knockouts, we could also do different knock-ins in case 9. Here I give two symbols and for this knock-in. The first is to knock in the construct with exogenous gene or like a reporter gene or overexpression of another gene at the 5' terminus of this gene's coding sequence, like downstream of the 5' untranslated sequence. And this will be stopped by a polydonation signal, like a transcription termination signal. So after the insertion of this through CRISPR Case system by homologous recombination, and then the allele will be looked at this. And this operating frame will be driven by the endogenous regulation sequence and expressed just as the endogenous gene, as the pattern of the endogenous gene. However, the transcription termination signal will then prevent the downstream sequence from transcription, and then that's disrupting the endogenous gene expression. And then in the other case, we could just design the target site at the downstream of the stop codon and upstream of the 3' untranslated sequence, and then design a donor like this, also containing this operating frame like a reporter gene or CRE or DRE recombinase. And then at the upstream of this operating frame, we will add a 2A peptide or ARIS as a linker sequence to make sure that the sequence downstream could be expressed after this integration of the donor. And then in this case, we can see that because there is no upstream transcription termination sequence, the expression of the endogenous coding sequence could be maintained. And because of the linker sequence like 2A peptide coding sequence or ARIS we will add here, the downstream operating frame could also be expressed. So this is a way of overexpressing another gene like a recombinase or reporter. And we could also maintain the endogenous gene expression in this manner. Last, let's talk about the transgene. So traditionally, transgene could be done in a random way by random insertion of the linearized plasmids, or even linearized back if we need to keep some regulatory sequence, or we do not make sure that where the gene is, or we want to simultaneously introduce overexpression of multiple genes. And then transposons have been applied into the generation of the random insertion transgenes like piggyback or other strategies, which is also well used in mice. However, in modern days, people now prefer more and more the super TG strategies via Cas9 or other endonucleases. Like we could just use homologous recombination to insert this construct like overexpression or other functions into the safe harbor in the mouse genome, like the H11, ROSA26, or other sites developing like Tegri or others. Then the advantage of this super TG compared to the random insertion is that the genotype and the insertion site of this construct is clear, and it is easy for us to do quality control of the animals we get, and to avoid the lineage construction, which will need additional generations and additional crosses which is generally more time consuming. Last but not least, we can also introduce point mutations by CRISPR-Cas9 system and generally a single-strand oligonucleotide donor. And we introduce the point mutation of interest in the exon and then design the CRISPR-Cas9 targeted site also at the exon we are interested in. And then after homologous-based repair, this donor which contains the point mutation will be then introduced in the target gene and generated the point mutation. We can see that since the disease-associated genetic variants are over 60% of them are single point mutations. So the strategy to generate a homologous point mutation in animal models like mice is being more and more important for the study of genetic diseases and rare diseases. So here's a summary of how the efficiency we generate the models by CRISPR Case in GemPharmatech facility. We can see in different project types like either conditional or conventional knockout or in different knocking cases or projects, we have a good birth rate range from like in the average of 12% or in 20% when generation of conventional knockout, which is the most easy one in these three. And we will calculate the goal germline rate different from the research papers that will directly calculate the number of positive founder mice with the number of embryos we inject at the beginning. We can see that in all the cases, the number is, range from like average of 2.5%, 2.4%, 2% to as high as a 7.5%, which is very high. So in this case, if we inject 100 of embryos, we can guarantee in most of the cases that we will get positive founder mice which have germline transmissions into the F1, and which symbols that the establishment of a stabilized inherited strain and the successful of the project. And then in next part, since we have a very good CRISPR-Cas9 based strategies and the technique, we launched this ambitious project in 2018, which is called the Knockout All project. So our goal in 5 years is to generate knockouts for all the protein coding genes in mice, like near 23,000 of them, and then simultaneously complete over 22,000 of conditional knockout alleles, like most of them are flux alleles. And during and then after we complete this project, we will become the largest mice source all over the world, and all the strains we have will have an independent property right. So from now on, we have already built over 9,000 of knockout lines and 8,000 of flux lines, and combine them together. We now combining other strains like point mutations and knock-ins and other tool strains. We now currently have over 19,000 of strains with independent property right, and now we are currently the biggest strain keeper all over the world. So why we could launch and efficiently do this knockout all project in our facility? We used a lot of automatic strategies, like we developed this software for the strategy design to generate conventional and conditional knockout strategy. And basically, we just input the basic gene information like gene ID into the software, and will automatically design the target sites and the donor sequence for us, which will greatly simplify the process we do the strategy development of the KOAP strains. And also we have developed other automatic strategy or softwares for the maintenance and the quality control of our strains. And also we have put many efforts into the classification of the strains to help the customers or researchers to better apply our models. Like we did the classification of our generated strains by gene families like GPCRs or zinc finger transcription factors or E3 ligases or whatever. And also we did this classification by a subcellular localization of the proteins, like if it is in the nucleus, if it is in the cytoplasm, in the mitochondria, or on the cell membrane, or others. And also we did this classification also by a tissue-specific expression or tissue-specific important function for people who study the mechanism or study the certain organ during development or during disease. And also then we have added all these informations into our search engine for our knockout all project strings. We can do this by gene name, strain name, by the tissues or organs we are interested in, or by different pathways or disease we are interested in. And also to use in combine with the flux strains we generate to finally achieve conditional knockouts, we have generated multiple tissue-specific Cray strains or inducible Cray strains, which means Cray ER strains in our facility. So by September in last year, we have generated and validated over 200 of Crays, which are mainly applied in the nervous system, cardiovascular system, and other important systems which will meet the broad interest of our broad customers all over the world. We have strict quality control standard of our newly generated Cray strains, since the precise expression of the Cray strains, which is the most important thing in the conditional knockout or conditional expression experiments. So the commonly used detection methods is the fluorescence metrograph or using frozen sections, or maybe we could use some LAC-Z-based staining methods to also do this, or we could also do HCF or easily just use Western blot to demonstrate the tissue-specific knock-in or tissue-specific Cray expression. So generally, we could cross these two strains together, one with a reporter strain, which has a ubiquitously expressed green fluorescence protein and the stop cassette, which will terminate the transcription, and this too is flanked by one pair of LOXPs. And then at the downstream, there will be another fluorescent reporter gene, like in this case, it's a T tomato red fluorescence. And it won't be expressed in the normal stage, in normal conditions, since the transcription termination cassette prevented from transcription. And then we cross this reporter strain with the Cray we want to test. And when we cross them together in their offsprings, in the Cray positive cells and tissue, the Cray will mediate the recombination of the LOXP sites, causing the upstream GFP and the transcription termination element to be deleted. And then the downstream reporter gene like T tomato could be expressed in a tissue-specific manner according to the expression pattern of the Cray, like in the Cray positive cells. So here's a real-world validation data of our PDX1 Cray. We know that PDX1 is very important for our pancreas development. So we can see that this green to red fluorescence change in most of the pancreas cells when we cross this PDX1 Cray line with our reporter line together. And also we can see some of the PDX1 positive cells in the antrostomach and in the duodenum. That's the validation we will give to most of the Cray lines we generate, and we will ensure that the Cray expressed in the tissues we desire, and give the support if there's some leaky or some not mainly expressed tissue. There's some expression in the not mainly conserved tissue to make sure the research data generated by our mice is, is of good credit. And we also have strict quality control standards for other mice generated during the knockout all project. We will do PCR genotyping and amplicon sequencing. And for the strains we generated by knock-in, we will sequence through the entire insert fragment in super TG or targeted knock-in. And then we will do quantitative PCR as well as other strategies to make sure that the strains we obtain will be single copy to verify the product and then apply it in our research or give it to our customers. And then we will do tests to avoid tandem repeat. So from now on, there's over 120 publications using the mice we generated through our knockout all project. Like there are conventional knockout lines, flux line, and Cray lines we generated. So here's a brief list of the knockout and flock strains or the CRE strains. We can see that there are some really of the high impact and the well-known journals here. So it means that our mice is well admitted by our academic customers and community. So in the third part, I want to give a brief introduction of the genetic disease models we tested in our facility. And we sincerely want these models could help the development of the rare disease drugs in our world. So the first one I want to introduce is the Duchenne muscular dystrophy, like the DMD. So the pathology of the DMD is that this mutation of this gene will cause a truncation of the protein dystrophy in our skeletal muscle cells and cardiomyocytes, which further leads to progressive skeletal muscle atrophy of the proximal extremities. And also during the development of this syndrome, the development of the central nervous system and other organs may also be affected, and that most of the patients won't live through 20 years old. So it is very severe. And also this disease have a very high prevalence in population of the world, like 1 in 3.5 thousand to 6,000 people. And the heredity of this disease, this DMD gene is an X-linked recessive. So let's briefly see one of the example of the etiology. In this case, 10 exons are deleted in this DMD gene. So if you are familiar with this gene, you know that this gene is really, really big. It have like near 100 of coding exons. So a simple deletion or simple frame shift mutation may cause out of frame dystrophin transcription. That is, it is truncated and this protein will be not functional. As we look here, this dystrophin actually acts as a bridge that combining our cell membrane and the actin fragments in our muscle cells together to make sure that contraction will work when our muscle needs strength. However, if the dystrophin itself is truncated, it will be dysfunctional and unstable in the complex and causing our muscles to be the function of a muscle to be disrupted. So currently, DMD treatment is generally done by gene therapy-based methods, like we can use exon-speaking methods by antisense oligos, like in BioMarin or in Sarepta Therapeutics, using like RNA interference or like morpholinos to do this. And also in these years, some pharmatech companies have developed this Cas9-based gene therapies, like in Pfizer and in solid belts and in the treatment, and it have already go into the third or the first or second phase. And we hope that these therapies could save the patients suffering from DMD. So in our facility, we generate this DMD mouse model by introducing this indel frame shift mutation at the 4th exon of the DMD gene in mice. And then this frame shift mutation will result in a truncation of the product of the gene product, which is the dystrophin in mice. And then after we obtain this homozygous, we did a lot of phenotypic analysis. Now I show just some of the data. We first test the graft strength of this DMD mutant mice. We can see that during the 16 weeks to 20 weeks, the graft strength is significantly lower in the mutant mice compared with the control. And also, we did this rotar test to test the strength of the muscle too. And then in the mutants, in both 14 and 18 weeks, the behavior in rotar test time is poor compared with the control group. So these are the functional tests of the muscles, and then we do some pathological tests in these mutants in 2 time points. One is 9 to 10 weeks, the other one is 22 weeks. We see the tricinimus and the soleus muscles confirmation. We can see that in the mutant mice, there's a typical maltrophy in these tissues, which is a classical symptom of the human DMD syndrome. And then some of the muscles functional disruption or can be observed in these sections. And also we test another important is the creatine kinase level in the blood of the mutant mice. And we can see that in the mutant mice, the blood creatine kinase level is significantly higher compared with the wild-type control. All this data suggests that the DMD mutant we generated is a good model to mimic the human DMDs. And another model I want to introduce in today's webinar is the hemophilia model. So hemophilia is, even more famous compared with the DMD. So the pathology is that the blood coagulation is dysfunctional in the patients because of the genetic defects of the factor 8 or factor 9. And these two defects are named as hemophilia A or hemophilia B, respectively. And between these two, hemophilia A accounts for about over 8% of the patients all over the world. Hemophilia patients undergo spontaneous or non-spontaneous bleeding, include difficulty to stop bleeding during trauma or surgery, as well as permanent joint damage. The prevalence is as high as DMDs, and it is a very large number, and it is also actually recessively heredited. And the general treatment of the hemophilia is the desmopressin acetate, which will help people to coagulate their blood when they are bleeding. However, this is just a treatment when we have this bleeding. We have this bleeding after we have this bleeding, and it won't be a permanent strategy to cure the hemophilia. So another companies like Spark or BioMarine have also developed the genetic gene therapies to overexpress the factor 8 or factor 9. In here, the ones I listed are mainly factor 8 to treat the hemophilia A by using adenosine-associated virus vectors to express this gene in human body to cure the hemophilia A. So we generated this hemophilia A mouse model by using this deletion strategy, designing two Cas9 sites, one at the exon 16, the other at the exon 70, to generate this small deletion in the F8 locus, and then causing this gene to be disrupted. And we can see that in the homozygous mice, the level of the factor 8 is decreased to nearly 1 to 2% of the wild type level in both male and female mice. And then when tested the bleeding weight and the bleeding time, and also the activated partial thromboplastine time in the F8 knockout mutants, and we can see that the bleeding is significantly more severe in the F8 knockout mice compared with the wild type control, which means that this model is a good model for the hemophilia study and the hemophilia drug in vivo treatment in mice. Oh, oops, sorry. And then in the last part, since today's topic is mainly focused on the genetic engineered mice, I want to briefly introduce about the humanized models, genetic humanized models. So first, I'll briefly introduce this animal humanization. So there's certain different scales of animal humanization. It can be from genes to cells to tissues and to environment. And all the purpose of these humanizations is to make our lab animal like mice to be a better stand-in for human research, like to make it more like human in genes, cells, or tissues, or the environments. So the different scale containing that gene humanization I talked before, and the humanization of the immune system, like we could introduce the peripheral blood mononuclear cells or the hematopoietic stem cells into immunodeficient mice to generate a human immune system in the mice. And or we could do this tumor humanization to test the human tumor or cancer cells behavior in mice, and then test the anti-tumor drug efficacy in mice by doing CDX or PDX first. And also we could do the microbiome humanization by using this GF mice and combined with human gut microbiome to generate this microbiome. or environmental humanized mice model. Since our today's topic is mainly focused on the gene modification lines, I'll focus on the introduction of the gene humanization strains. So typically in our facility, we use these two strategies to generate most of the gene humanization lines. The first is to humanize the whole gene by introducing this human coding sequence at the 5' of the wild-type mice homologous gene in the methods I introduced before. And then this construct was terminated by this polyadenation signal transcription termination. And then the expression of the endogenous mice gene will be substituted by the full-length whole human homologous gene, which is called whole gene humanization. And in another case, if we don't need the gene to be full humanized, or we need some of the gene to maintain in the mice stage, we just substitute this mice coding sequence of the part we want to change by using 2 CRISPR Case target sites to delete this sequence, and then using a donor to replace these exons by the human coding sequence of this homologous region. And then we'll generate a chimeric allele expressing the chimeric human mice gene, which is called the partial gene humanization. This strategy is particularly valuable when we study membrane proteins or like immunocheckpoints that we substitute the extracellular domain of this protein for research like for affinity research or the drug efficacy test of the monoclonal antibody drugs. And then we maintain the intracellular domain and the transmembrane domain to keep the downstream signal transduction pathway in mice identical and see the mice response like the treatment response and toxicity response. So currently using these strategies, we aim to develop more than 700 of the humanized strains, and now we already have over 300 of them. And for the most of them are the humanized immunocheckpoint mice strains. We developed the single target strains and then cross them together or generate new humanizations based on these strains to get double target and triple target strains. The one in bold here are the strains we already have, and the one not in bold here is the strains we are currently developing or crossing together. And also in most of the strains, we developed them in 2 different genetic backgrounds. One is the commonly used Black 6, and the other is the BioBC. So why did we do this in different backgrounds, and how we keep to do them in multiple strains? And here's a elegant drug efficacy test in this study by using a combination therapy by the anti-PD-1 and anti-TIGIT antibody in different backgrounds responsible to breast cancer cell lines. Like we can see that in the left, when in the BioBC, humanized PD-1, humanized TIGIT bearing this CD26 lines with the therapy of this anti-TIGIT based on the anti-PD-1 effect. In this, the tumor growth inhibition is about near 80%. However, if we use this B6 double humanization strain bearing the MC38, bearing the MC38 cell line, we can see that the tumor growth inhibition is not significant compared with the vehicle treatment when we use the anti-TIGIT antibody. So why? To further study the reason, we analyzed the tumor infiltration lymphocytes in these 2 studies. We can see that compared with the vehicle treatment, the NK cell level is not significantly higher in the B6 background study. And also, what's worse, the Treg cell is increased in the lymphocytes in this B6 background. We know that Treg cells will inhibit the cytotoxicity of the NK cells or the CD4 positive, the CD8 positive cells in our microenvironment, and then just preventing these drugs to be efficiently clearing to mediate the ADCC effect and cause anti-tumor efficacy. However, the things are totally different in the BioBC background experiment. We can see that compared to the vehicle control group, the level of the NK cells and is increased dramatically after treatment, and also the Treg cell is decreased, which means that which could partially explain why that BC background humanization model is more suitable for the drug efficacy test, and the tumor growth inhibition is better in this strain. And also on the other hand, we tested the toxicity in both backgrounds using the combination treatment of anti-PD-1 and CTLA-4 together in both backgrounds. We can see that in this toxicity experiment, the BC PD-1 CTLA-4 humanized mice reproduced the phenotype of this joint swelling and the skin inflammation we can see here, observed in clinical practice, which have been already demonstrated in clinical trials, which means that different backgrounds may help us to better explain the toxicity, to examine the toxicity and the drug efficacy in the preclinical in vivo tests. Also, the other model strains I want to introduce is that the humanized AC2 mice, because the COVID-19 pandemic is still, wild all over the world. We believe that these strains will greatly help the anti-COVID-19 research in our society and all over the world. We have generated multiple humanized COVID-19 receptor ACE2 lines in different backgrounds, like in B6 background, chimerical full-length ACE2 mice, or ACE2 overexpression mice using a K18 promoter, which is mainly expressed in the epithelial cell of our lung, and also in the BC background or in the severe immunodeficient NCG background. So we have already examined all of the models we generated here. We examined the expression of the model, the expression of the humanized ACE2 in our model, and then we test the behavior and the physiological phenotype after infection of these mice. We know that if we don't humanize this ACE2, the COVID-19 won't infect the wild-type mice. However, in our humanized mice, we can see that after infection, in this first several days, the body weight of the mice decreased dramatically, and then after treatment or after its self-recovery, the body weight gradually recover and increase. And then we also test the virus loading in the lung in this B6 humanized ACE2 mice models. At 1 day or 3 days post-infection, we can see that the virus load in the mice lung is significantly higher. And then we also test the neutralized antibody in the blood, and all these things demonstrate the expression and the functionality of this humanized ACE2 mouse model for study of COVID-19 infection in mice. Also, our different mice have been applied in multiple mechanism study or drug discovery studies against COVID-19. And here's one example that is developed in collaboration with the West China Medical School, Sichuan University, and the CAS Kunming Institute. They developed this drug, MI9 and MI30, for anti-COVID-19 efficacy test, and using our B6 humanized ACE2 model. We can see that this MI9 or MI30 in different doses after infection, which will greatly lower the total RNA copy number of the infected virus, which means that our model is suitable for the preclinical drug selection and drug evaluation of the anti-COVID-19 drugs. We sincerely hope that our models will greatly help this kind of research and to terminate the pandemic all over the world as soon as possible, if we could contribute our power. So last, I want to briefly introduce about our company. As we can see that this is the data collected by MSR. So exclude the embryonic stem cell lines of mice, there are currently more than 79,000 mouse strains all over the world. And among of them, nearly thousands of strains are developed by GemPharmatech with independent property right. And now we can confidently see that we are the biggest strain keeper all over the world, and we are the biggest strain keeper and the strain commercialized company all over the world. And we provide one-stop solution for biomedical research for our customers, both in the scientific or in industrial field. We can offer conditional knockout lines generated by our KOAP project, humanized strains by our drug screening strains, and then we can also offer immunodeficient strains and some of the genetic disease strains. And also we could do customized model generation according to the need and the design of our customers. And also in most of our mice, we could give a germ-free options, and we also sell germ-free mice in regular strains like B6, BioBC, or immunodeficient NCG strains. And also we provide the listing services like mouse breeding and cryo preservation of the important strains. And we also do preclinical drug efficacy and phenotyping study and tests in our facility. And also we could help you to do mouse genetic resource import or export for full service for the rapid delivery. So altogether, we aim to give our customers a one-stop solution for biomedical research, especially for the basic research and the preclinical drug studies. Okay, that's all. Thank you for your attention today. Your question is warmly welcomed. Thank you again. So if you have any questions, please feel free to type your questions in the chatting dialogue, and I'll give my answers if I could. Okay, here's a question coming, just how to validate the efficacy of pre-medicated conditional knockout. So yes, this is a good question because it is very important to, like this is a quality control of our real research. So for conditional knockouts, the most important thing is that we need to set control groups, like using Cre positive and Cre negative strains, and use Cre positive strains with homozygous or heterozygous flux alleles, and to study the the expression level of the gene of interest in different tissues. And also because we also did the Cre validations I introduced before, we are confident that the expression will be in the correct tissue if we do the analysis. And then we need to, if we have different methods to sort these cells or tissues by, either we just take the organ out or just we use sorting using flow cytometry or other methods, and then we can evaluate the relative mRNA level or even protein level to see the efficacy of the pre-medicated conditional knockout. And now we have this service doing the mRNA level test of our strains of conditional knockout or conventional knockout. And also we are thinking that if our customer really need it, we could do this protein level analysis service as well in our facility. Yeah. Is it necessary to generate gene humanized models? Oh, yeah. Actually, it's not necessary, but it is recommended, since these methods will, express the exogenous human gene in the same tissues or organs in the same pattern in mouse, as this target knocking will be controlled by the endogenous regulatory elements in the gene. But however, in other cases, if you want it to be expressed like in a human manner, like using humans, regulatory elements, you could just introduce a human back by a random insertion or by a targeted methods. And then you could either knock out the endogenous mice gene if you need, or if you don't need it, you just need a humanized marker for your further study, like you need to use it as a marker of the certain disease or certain cell types, you even don't need to, knock out the endogenous homologous gene. So it is not a necessary thing, but it is, in most of cases, it is recommended. Yes. Mm. Mm. What's the difference between different AC2 mice models? Yes, the first thing is that why we need to develop it in different backgrounds. The reason of that we develop this in both B6 and BioBC is that we already see the difference of the immune system in the PD1 and the CTLA4 or TIGIT humanized models before. The response of the drug efficacy or drug toxicity of the same drugs in different strains can be different according to their basic difference of the immune system. So we generate them in the both lines. And for the NCG lines, we want to use it in combination of the immune system humanization to further study like the side effects or the cytokine storm, the positive cytokine storm during infection and the drug treatment. And also the difference between, the humanization by knocking or humanization in the H11 locus in a K18 driven overexpression manner is that this K18 overexpression is with a larger expression amount and the tissue is mainly focused in the lung. So the infection will be more severe compared with the targeted knocking in the mouse AC2 locus. So it is better for the study of the severe COVID-19 infection. And in the other case, it's better for the, general or moderate infection study. Okay, so since our time is near the limit, so I'll just wait here for another minute to see if there's other questions or other interest by our audience. Okay, because we see that there's no more questions from our audience. So thank you all a lot for attending this webinar today. I hope that we can have further collaborations between each other. And if you have any further questions or comments or suggestions, please feel free to contact us when you are convenient or you want. Okay, thank you. Thank you. Okay, goodbye. We should have a good day or good night.